The overall goal of the following experiment is to generate transgenes using worm genomic DNA carried in a fosmid library clone. The use of genomic DNA preserves all of the native control elements, such as enhancers and 3' UTR regions. This is achieved by moving the fosmid into the SW106 E. coli strain to utilize the inducible lambda red recombination machinery. As a second step, the galK gene is inserted into the desired location in the fosmid, which marks the site where the GFP or TAP tag will be placed. Next, the galK gene is replaced by GFP or a TAP tag via homologous recombination, followed by negative selection using deoxygalactose in order to create the modified transgene. Results are obtained that show these transgenes can be created quickly and successfully, based on verification of tag insertion by colony PCR.
Hi, my name is Kashyap. I work in the Fisher Lab at University of Pittsburgh. Today, I'm demonstrating the technique for making elegans Transgene by Recombineering with galK as the selectable marker. The main advantage of this technique over existing methods of creating elegans Transgenes is that in this technique, we use the genomic DNA of the gene of interest and thus are able to cover all the critical elements of gene regulation, such as control elements in 3' UTR region, alternative splice transcripts, alternative promoters, and distal enhancers, which might be missed out in conventional standard cloning-based approaches. So let's get started.
Order the fosmid clone for the gene of interest, or GOI, from Gene Service using Wormbase as a guide. When selecting clones, we choose ones that have the